Dear students, this is ninth biology video class, unit 18, organization of tissues. Unit 18, organization of tissues, part 9. Unit 18, organization of tissues, part 9. Last class, we have seen what is cell division, the types of cell division we have seen, what is amitosis we have seen, and also we have seen events of mitosis we have completed. The next topic we are going to see is the one meiosis. What is that one? Meiosis. The term meiosis was coined by scientist name called Farmer, the year of 1905. It is a kind of cell division that produces the sex cells or the gametes. So meiosis is the type of cell divisions. They produce sex cells, or we can also say as a gametes. It is also called as reduction division because the chromosome number is reduced to apply n from diploid to n. The chromosomes, we have 46 chromosomes. That 46 chromosomes, when it is transferred to the next generations, it will be in a half number. That is a 23 chromosomes. So half number, that is we say as a single n, and become as a 2n means diploid. Father and mother chromosome joined together, they become as a diploid one. Meiosis produces 4 daughter cells from a parent cell. We'll see what it is. Meiosis consists of 2 divisions. They are heterotypic division or first meiotic division. The next division is the one homotypic division or second meiotic divisions. We'll see one by one. First one is the one heterotypic divisions. Heterotypic divisions. What does that means? Heterotypic division means it divides a diploid cell into 2 haploid cells. So diploid cells means 2 cells, that is because a diploid cells, they become as a 2 haploid cells. The 2 cells will become as a single cell, that is they say as a 2 haploid cells. The daughter cells resulting from these divisions are different from the parent cell in chromosomal number. That is because a heterotypic. The daughter cells resulting from these divisions different from the parent cells in the chromosome number. That is because a heterotypic. This consists of 5 stages. This heterotypic consists of 5 stages. Just keep in mind. We'll see one by one. I'll just read it out. Listen carefully. Prophase 1. Then B, metaphase 1. Then C, anaphase 1. Then D, telophase 1. Then E, cytokinesis. So these are the 5 stages of heterotypic divisions. We'll see one by one. What is prophase 1? What is prophase 1? Just I'll show you the diagram where you can understand better. Take your diagram here. So this is what the diagram of meiosis 1. As you have seen events of mitosis, here also the events of meiosis, they given the diagram. First one, interphase. First one is the one interphase. As we have seen the mitosis, That is a resting phase, we can say, or getting ready for the cell divisions, or taking rest, we can say. That is because of interphase. Then next event is the one prophase 1. Next event is the one prophase 1. What is prophase 1? Here, the prophase 1 takes a longer durations. Prophase 1 take longer durations and is subdivided into 5 stages. Subdivided into 5 stages. They are leptotin. Just repeat after me. They are leptotin, zygotin, pacatin, diplotin, diakinesis. Diakinesis. So these are the types of prophase 1. That is a longer durations. The 5 stages of prophase 1 is the one. Again, just repeat. We'll see one by one. I'll show you in the textbook and just read where you can repeat after me. Where you can repeat after me here. This is the one. Prophase. This is the heterotypic. Prophase 1. In the prophase 1, leptotin. Repeat after me. Zygotin, pacatin, diplotin, and diakinesis. We'll see one by one. What is leptotin? The chromosomes become uncoiled and assume long thread-like structures and take up a specific orientations inside the nucleus. They form a Bowkit stage. They form Bowkit stage. That is zygotin. Adjoining. Zygon adjoining. Adjoining means just come near. Zygotes, they come near. The chromosomes come near. That is, this as a zygotin. Two homologous chromosomes approach each other and begin to pair. The pairing of homologous chromosomes is called as a synapsis. Pairing of homologous called as a synapsis. So, this is the information about leptotin. Leptotin. The next one is the one zygotin. Next one is the one zygotin. We have seen. Then pacatin. We have just what do they say here? Zygotin. The chromosomes, as you see chromosomes here, these chromosomes, they'll come and contact each other. They'll come and contact each other. That's what they say here. Zygotin. And pacatin, what they say pacatin? Pacus means thick. The chromosomes are visible as long pair twisted threads. The chromosomes visible as long pair twisted threads. The pairs so formed are called bivalent. The pairs so formed are called bivalents. Each bivalent now consists of 4 chromatids. That is, we call it tetra stage. Tetra stage. The homologous chromosomes of each pair begins to separate. They do not completely separate, but remain attached together at one or more point by X-shaped arrangements known as chasmata. Here, this proposed one, prophase 1, the both end of the chromosome, they'll just go in contact. That is, they say as a synapsis. Already we have seen zygotin. A chasmata means that together, that remain attached together. A more point will be attached to the X shape. That is, they say as a chasmata. They'll ask you 2 marks. The chromatid breaks at this point and broken segments may be interchanged. As a result, the genetic recombination takes place. The next one is the one diplotin. Next one is the one diplotin. Here, diplotin, each individual chromosome, here, I'll show you the explanations. diplotin. Here, each individual chromosomes of each bivalent begins to split longitudinally into two similar chromatids. What they say here, the chromosomes are bivalent two that arms like structures. They begins to separate. That's what they say as a split in longitudinal directions, the straight directions into two similar chromatids. The homologous chromosomes repel each other and separate. The chasmata begins to move along the length of chromosomes from the centromere towards the end, resulting the terminalization. Towards end, resulting in terminalizations. The next one is the one diakinesis. The paired chromosomes are shortened and thickened one. The nuclear membrane and nucleolus begins to disappear. The nuclear membrane and nucleolus begin to disappear. Spindle fibers makes their appearance. The next phase is the one metaphase 1. Next phase is the one metaphase 1. Here the chromosomes move towards the equator and finally they orient themselves on the equator. The two chromatids of each chromosomes do not separate. The centromere does not divide. Here, this is what the metaphase. I'll show you the diagram. Metaphase 1. So the chromosome moves toward the equator. Finally, they orient themselves on equator. They'll just move to the central part of that cell. That is, they say as an equator. The two chromatids of each chromosome do not separate. What about anaphase? Next stage is the one anaphase. What happens in anaphase? We'll see. Each homologous chromosomes with its two chromatids and undivided centromere move towards the opposite poles of the cell. This stage of chromosomes is called as a diet. This chromosome is called as a diet. And the next one is the one telophase. Here, the applied number of chromosomes after reaching the respective poles become uncoiled and elongated one. The nucleus membrane and the nucleolus reappear and thus two daughter nuclei are formed. Then next stage, the meiosis 1 stage, cytokinesis. Here, cytokinesis, the cytoplasmic divisions occurs in two applied cells are formed. Here, this is what the cytokinesis. You can see two applied cells are formed. The telophase is become as a next events of cytokinesis. Here, two applied cells are formed. And so far we have seen heterotypic divisions. The prophase is the one long durations and they have leptotin, zygotin, pacotin and diakinesis. Then metaphase 1, then anaphase 1 and telophase 1 and cytokinesis. So these are the heterotypic divisions. The next one is the one homotypic divisions. Here the homotypic divisions, can you all see here? This is what the meiosis 2, you can see. It is the divisions that two applied cells formed during first meiotic divisions divide into four applied cells. So they divide into four applied cells. Can you see here? This is what the four applied cells will be formed here. The daughter cells are similar to parent cell in the chromosome number. It consists of 5 stages: prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. We'll see what it is one by one. We'll see. First one is the one prophase 2. Here, prophase 2. So, interkinesis, interface, we have interfaces here, interkinesis takes place here. The prophase 2, the centrioles divide into two. Each one moves to opposite poles. Aster and spindle fibers appear. Nuclear membrane and nucleolus disappear. Then metaphase 2, here the chromosomes get arranged on equator. Chromosomes get arranged on equator. Okay, then two chromatids are separate. Two chromatids are separate. The next one is the one anaphase 2. Anaphase 2. Yeah, this is what the anaphase 2. Here, what they say here, the separate chromatids become daughter chromosomes and move to the opposite pole. The next phase is the one telophase 2. Here, the daughter chromosomes are centered. The nuclear membrane and nucleolus appear. The next event is the one cytokinesis 2. Cytokinesis 2. Two cells are formed from each aploid daughter cells, resulting in the formations of 4 cells with aploid number of chromosomes. The next one is the one significance of meiosis. Next one is the one significance of meiosis. Here, this is the significance of meiosis. The constant number of chromosomes in a given species is maintained by meiotic divisions. The constant number of species will be maintained, the chromosomes. The genetic variations is produced due to crossing over within the species, which is transmitted from one generation to next generations. Okay, then this is the significance of meiosis. The next difference between mitosis and meiosis. So mitosis is important. 4 marks can be asked. Mitosis occurs somatic cells. Meiosis occurs reproductive cells. Mitosis involves the growth and occurs continuously throughout life, whereas meiosis involves gamete formations only during reproductive active age. Consists of single divisions. Consists of 2 divisions. The 2 diploid daughter cells are formed. 4 haploid daughter cells are formed. The chromosome number in daughter cell is similar to the parent cell, that is 2n. The chromosome number in daughter cells is just half, that's a 1n of the parent. Identical daughter cells are formed. Here, the daughter cells are not similar to the parent cell and are randomly assorted. So these are the difference between mitosis and meiosis. This is very, very important one. Learn today itself. If you have any doubt, you can contact me through WhatsApp. Okay, bye. Take care.